Hello everyone. Today's video is about polysaccharides. So as we already know that polysaccharides are a class of carbohydrates, and in carbohydrates we have already studied that the carbohydrates can be of three types. These can be monosaccharides, these can be oligosaccharides, and these can be polysaccharides. In the previous videos, we have already discussed about monosaccharides and oligosaccharides. So in today's video, we'll be mainly focusing only upon the polysaccharides. Now, what are polysaccharides? These are a class of carbohydrates which upon hydrolysis will give us more than 10 units of saccharides or sugar. And the most basic one is the glucose. So it will give us basically more than 10 units of glucose upon hydrolysis. Now, these polysaccharides are the largest group of carbohydrates, and these polysaccharides can be further classified into either homopolysaccharides, or these can be classified as heteropolysaccharides. Now, what is the difference between these two? Homopolysaccharides will be made up of only a single type of saccharides. For example, it will be made up of only glucose unit, glucose, and so on. Heteropolysaccharides will be made up of different kind of saccharide units. These can be either made up of galactose, and so on, and also other repetitive units. So homopolysaccharides are those polysaccharides which will have a single type of sugar. For example, we have starch, we have cellulose, etc. And heteropolysaccharides can be either agar or pectin, and so on. So we'll be majorly discussing about starch, cellulose, glycogen. What are these? What are their functions? And what is their structure? How do they look chemically? So let's start with that. So the first one that we'll be discussing today is the starch. Let me start with that. Starch. The first polysaccharide is starch. Now, what is starch? Starch, we all know, is a complex carbohydrate, but it is made up of a single type of sugar. The single type of sugar is glucose. Starch will be made up of a number of glucose units, more than 100 units of glucose units. Now, we know that the structure of glucose is like this. It is a 6-carbon structure. So this is the basic structure of glucose. We have 6 carbons here. This is carbon number 1, this is carbon 2, this is carbon 3, this is carbon 4, this is carbon 5, and this is carbon number 6. This is a basic unit of glucose. All the structures that we'll be studying today are all made up of repetitive units of glucose. So this is a very basic structure that you must all memorize. This is called as the alpha glucose. Alpha glucose, we have already discussed about the alpha and the beta forms, but today we'll be specifically studying all the structures that are made up of alpha glucose. Now, this starch is a homopolysaccharide. It is a homopolysaccharide, means it is made up of only a single type of sugar, which is the glucose, and it is a storage carbohydrate of plants. In the root, stems, seeds of plants, there is starch. Also, the potato tubers, there are different cereal grains. They are also very rich sources of starch in the human diet. A very important thing is that the starch is one of the carbohydrates which is digested in the mouth of humans, first of all. In the mouth, there is an enzyme called amylase or ptyalin, which breaks down the starch. Now, how does a starch break down? See, a starch is made up of linear chains of sugar like this glucose molecules. We will have more than 100 units of glucose which are bound together by alpha 1,4 glycosidic linkages. We have already discussed about what are alpha 1,4 glycosidic linkages, but I'll give you a brief review about that in the coming diagrams. For example, this is a linear chain of starch, more than 100 units will be here. Now, when amylase will act upon the starch chain, now the starch will first break down into maltose. Now, what is maltose? Starch is made up of more than 100 units of glucose. When amylase will act upon it, this starch will break down into maltose. Now, maltose is made up of 2 units of sugars. It is made up of 2 units of glucose. Further, on this maltose, another enzyme will act, which is called as maltase, and this maltase will break down the maltose into single units of glucose. In this way, a complex starch chain is broken down into simple units of glucose, and this glucose is readily absorbed by our body. So this is the structure of starch chain. When upon the starch chain, when amylase or ptyalin will act, it will break down into maltose, which are only two structures of which are made up of two structures of glucose, two subunits of glucose, and further on this maltase will act, which will break it down into different single units of glucose. This is a single unit of glucose. Now, how does maltose form? This I have already discussed in detail in my previous video. So for that reference, you can just go back to that video. Next, this starch, it is also of two different types. The two different types of starch are amylose, amylose and amylopectin. Now, I would like to mention here that amylose and amylopectin are also made up of repetitive units of alpha glucose only. Both of them are made up of repetitive units of alpha glucose. But the only difference is that amylose is a single linear chain, whereas amylopectin is a branched chain. Now, let me show you a diagram for this first. First of all, let me show you a diagram for this one, for the starch. And then we'll see the diagram for amylose and amylopectin. So here we have, this is the structure of starch. Here you can see a single glucose molecule. Here it is bound by alpha 1,4 linkages, and this linear chain, when this is formed, this is called as the starch, which is the polysaccharide. Now, let me show you this amylose and amylopectin. So here you can see this is the amylose and this is amylopectin. Now let me first explain this diagram and then we'll discuss about their properties. Here you can see this is a single alpha glucose unit, another alpha glucose unit, another alpha glucose unit. This is also glucose unit. So more than 100 glucose units are also present in this amylose. Now, as you can see in the glucose, simple glucose, this is carbon 1. Just a second. This is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5 and carbon 6. Similarly, here we'll have oxygen, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5 and carbon number 6. Now, you can see that in amylose, these two glucose units will come together and a water molecule is lost from carbon number 4 and carbon number 1. That is why this is called as alpha 1,4 glycosidic linkage. Now, when a single linear chain of this glucose molecules is formed, this is called as amylose. Now, amylose, it's an unbranched chain. It is a linear chain of glucose, and these are joined by alpha 1,4 linkages. This is soluble only in hot water. It won't dissolve in either very cold water or water at room temperature. It will only dissolve in very hot water. Now, coming to amylopectin, now again, if you see this structure of amylopectin, this one is similar to amylose. Means we'll have this carbon 1, carbon 2, carbon 3, carbon 4, carbon 5 and 6. This carbon 4 and this carbon number 1 will lose a molecule of water, and that is why this linkage is alpha 1,4 glycosidic linkage. Similarly, we have another chain. Now, in amylopectin, these two starch chains will also bind together in a linear way, in this way. So, carbon 1 from this glucose will join with carbon number 6 of this glucose molecule, giving rise to a branch structure which is called as alpha 1,6 glycosidic linkage. So basically, amylopectin is similar to amylose. It will have two or more than two amylose chains, which will have an additional alpha 1,6 glycosidic linkage. And this chain-like structure, this branched structure, will give rise to a very complicated, to a very complex carbohydrate structure. Now, let me show you a picture for this, for amylose and amylopectin. Now, this is amylose. This structure is amylose. You can see we have alpha glucose, alpha glucose, alpha glucose, alpha glucose, and so on. And in between carbon 1 of this and carbon 4 of this glucose, there is a bond of a linear chain, which is called alpha 1,4 glycosidic bond or glycosidic linkage. Similarly, let me show you a diagram for this, for amylopectin. So this is the structure of amylopectin. So here you can see this is, if on a basic level we see this is one chain of amylose, this is another chain of amylose. In amylose, the glucose are joined by alpha 1,4 linkages. Now, additionally, this carbon number 1 of one glucose and carbon number 6 of another glucose will also form another bond, which is called as alpha 1,6 glycosidic bond, and this linkage will lead to a very branched structure, due to which amylopectin is also called as a complex carbohydrate, which is very difficult to break down. So I hope this is clear with everyone. Now, coming back to the properties of amylopectin, amylopectin is totally insoluble in water. It's totally insoluble in water. Now, there is a test for difference between the starch and the glycogen. Now, first of all, let me discuss what is glycogen with all of you, and then we'll study about the chemical test that can differentiate between a starch and a glycogen. So here we have the structure of glycogen. Now, again, if you see, now again, this glycogen is made up of alpha glucose, repeated units of alpha glucose. The difference between starch and glycogen is that a glycogen is a very, very, very complex form of starch. It will not have just the branch chains, it will have more than 1,000 branch chains per molecule. The structure of glycogen, I'll show you a clearer picture for that, but first of all, let me explain. So here you can see, again, we have this alpha 1,4 linkages, alpha 1,4 glycosidic linkages, and here you can see this is carbon 1, this is carbon 6, so we'll have alpha 1,6 glycosidic linkages here as well. This glycogen is called as the animal starch because in animals or in humans, most of the energy of which is stored in the liver is in the form of glycogen only. It is a storage carbohydrate of animals. Now, it is also homopolysaccharide because it is only formed of repetitive subunits of alpha glucose. Now, the most important thing is that the structure of glycogen looks similar to amylopectin. The only difference is here, I'll show you. The only difference here is that here you can see This is a linear chain, alpha 1,4 linkages. This is another chain that is branched out from the main chain, which is joined by alpha 1,6 linkages. Now, if we see it on a bigger level, this is an entire diagram uh, of glycogen. Here you can see this is first linear chain. The linear chain will have one branch structure. This branch structure will further have another branch structure, and this branch will further have another branch. So when there are branches of the branches, means when the complex structure is further overcoiled upon itself, it will give rise to the glycogen. So glycogen, being so complex, is basically the storage material for the higher organisms like the animals or the humans. Now, there is a test to differentiate between starch, amylose and amylopectin, of course, between starch and glycogen. Now, this test, it is called as the iodine solution test. If we pour iodine solution on a starch, either amylose or on amylopectin, then starch reacts with this iodine solution and gives blue color. Whereas when this iodine solution is poured upon the glycogen, it will give us red color. So this is a basic chemical test to differentiate between starch and glycogen because they have almost similar structure. The only difference is in between their complexity. So I hope the structure of starch and glycogen, amylose, amylopectin and glycogen, and the polysaccharides, part 1 of polysaccharides, is clear with everyone. In the next video, we'll be discussing about the structure of cellulose and chitin in detail. In case there is anything else that you need a detailed explanation of, you can always let us know. You can always write to us. Your feedback is most welcome. Thank you for watching the video.